Okay, so our next session is going to be focused on population metrics as indicators. So the notion is that those adults came from somewhere. They came from a population, and that population can be monitored at a number of different life stages. And so the first life stage we're going to be talking about is freshwater outmigrant, and Zimmerman from WDSW is going to be talking.
So I have to say that when I came to the Pacific Northwest in the fall of 2008, I've been here for about 2 months, and my supervisor at the time came to me and she said, I need you to produce a forecast of coho run size for all of Washington State. And I said, okay. And she said, and here's the document how they did it last year. And she walked away. And I said, okay. So that's all a way of saying that I think that the conversations that I have desired for 4 years are starting to come to pass. Just in this week, talking with you all here, looking at this question from an ecosystem perspective, having the stock assessments, the ecological, the ecology aspects of it, and the physical oceanographic processes. And so I think this has been a really productive and fruitful productive week. So my task here is to talk about freshwater monitoring of Puget Sound salmon. And I started to put together a list of all the people that have contributed to data points that I am presenting today, and it was quite a long list. So clearly there's a lot of people who have contributed to this work at the Washington Department of Fish and Wildlife, and a lot of funding sources that have made this work possible. And that's my sole point on this slide. So I'm going to talk a bit about the monitoring framework that we had in place in Puget Sound, and then talk about the what, where, when, and how of this framework, and then applications to forecasting sort of through my lens, which is rapidly, hopefully, improving this week. So the monitoring in freshwater is actually 2 parts, right? It's the fish that come back, the spawner, and then the fish that go out, the smoke. And both of those measurements are taken in freshwater, and through this then we can partition the survival that occurs in the freshwater from the marine environment. And as we all know, there are very different ecological processes that occur in these 2 environments. So when we're talking about forecasting, we're interested in the number of fish that go out, and then we're interested in making a prediction as to what the run size will be somewhere around here, if this is sort of a timeline when they recruit into the fishery, and before they come back to spawn, so that we can set our fisheries and plan our fisheries appropriately. So this idea of looking at life cycle monitoring was actually formalized into a fish in, fish out framework, and this was developed in 2007 to guide salmon population and habitat monitoring statewide. However, super useful if we're working on the topic of forecasting, which is the subject today. So this map here shows for Washington State where we operate smoke traps in Puget Sound, and smoke traps are often the limiting factor when you're trying to set up a life cycle monitoring program because they're expensive. They take a lot of staffing and a lot of equipment. There are many other smoke traps that are operated in Puget Sound by different tribal and non-governmental entities, and so that would add to this list. But I focused just here on the ones that are operated by the Department of Fish and Wildlife. So we measure the number of fish out. If it's a small creek, we might have a expanding weir. If it's a larger river, then we have a smoke trap that we would set up a mark recapture study design to estimate the number of smokes that move out each year and their timing and their body size. And then when it's a really large river, we still try to do this. This is the Skagit River trap that's been operating since 1990 with a fair amount of success, which you might not necessarily think looking at how tiny it is with respect to the river, but it's part of it is just a challenge of where to place the trap in the river so that you can set up your mark recapture study design. So there are also estimates of spawner abundance, so the other side of the freshwater monitoring. which we accomplished through counting live fish, dead fish, number of reds, and accomplished this in collaboration with many others. I would note by foot surveys, aerial surveys. In some cases, we have tracks and weirs. Those are the best data because they're our hard count in that way. And then more recently, we've been working to improve the Chinook escapement estimates in Puget Sound in a select number of populations using a genetic marker capture tool as sort of an experimental tool to get unbiased and precise estimates of Chinook spawner escapement. So if we take all these smoke tracks and put them together with the adult monitoring, we have life cycle information for 7 different Chinook populations or subpopulations, 13 coho, 5 chum, 5 pink, 2 sockeye if we're looking just from spawner to fry and fry to spawner, and then 9 steelhead. So there's a fair amount of information, and then the amount of information varies among species as well. So the juvenile traps are operated from January through August. It depends on the watershed. This is the Dungeness River where the smokes don't really start coming out until early February. So we start that trap a little bit later. Chinook, the length of the trapping season is really dictated by the Chinook outmigration because they take the longest to leave the system. They seem to have the greatest life history diversity in terms of the way in which they're using the freshwater environment to rear. So this blue line for Chinook, Chinook tend to come out in a cellular Chinook in a bimodal. There's a fry migration early in the season, which sometimes is a lot higher. This year there weren't very many fry coming out of the Dungeness. And then there's a par migration. These are fish Chinook that have reared in freshwater, cellular Chinook that have reared in freshwater for more extended periods of time, 3 or 4 months, and they're 90 to 100 millimeters by the time that they're out migrating from the system. Then pink and chum tend to come out in sort of early April there, but fall chum, summer chum populations would come out a little bit earlier. Steelhead and coho, 3rd or 4th week in May. And you can see, though, that's not always consistent. So this green here, that's the coho. And then this year in the Dungeness River, there was an early pulse of coho that came out as well. So there are some life history variability there as well. We put all this together for each watershed. This is just an example from the Dungeness River in 2012. We can stack the production of all the species that come out. And this was a year where we had Ivan toucans spotting the year before, and so there's quite a few pink fry outmigrants. So this is the type of information collected through the juvenile or smoke trap monitoring component of the freshwater monitoring. If you are interested in more information on the juvenile monitoring, we do have a web page that's posted on the department's website, and actually later, this is a really long web address below, but we do have reports and production estimates and all that available. Okay, so the next part of my talk here is how would this life cycle monitoring information apply to fish conservation and management. And I see 3 major applications, and I'm going to focus in obviously on the forecasting application. One is to derive biological reference points, and there's been some discussion here about how spawner recruit data is specific slaughter. But when you divide it up between the freshwater and marine environment, that's not necessarily the case. And there is some information on watershed capacity and the spawner to produce maximum sustainable yield that you can obtain. The second application is to identify the life stage that is limiting the productivity of stock. And then the third is to predict production of the desired point of the life cycle, and that's the run-side forecasting that we're discussing today. So this is that life cycle again that I started with, and while we can develop all kinds of complicated models to predict run-side, what we're really talking about is the smoke and the marine survival equals a certain number of recruits. So we might have different ways of estimating smoke or predicting smoke, different models that predict marine survival, but ultimately it all kind of boils down to this, and somehow this is easier for me to get my head around, which is why I pick here rather than the long equation. I'm going to talk first about the smoke and how that might be applied to forecasting and how we currently apply it to coho forecasting. If we can measure smolt, that's great. That's our best quality information, particularly if we've set up our study design correctly to make that abundance estimate. But we can't put a smolt trap in every river, and we are trying to estimate total or predict total run size, so we have to extrapolate what we know from systems where we do measure it to systems where we don't measure it. And that is one of the major challenges on the freshwater side that contributes to the run size forecasting. Oops, that's the wrong button. All right. So I would argue that one of the things that we need to understand as we're setting out to figure out, we're talking about the freshwater side here right now, is the role of density-dependent and density-independent functions in terms of how these fish are behaving in freshwater, how the production coming out of freshwater, what is it that is affecting them, and this is why I say that. If you have a density-dependent function, like this Robertson-Holt curve on the left there, we would expect for low numbers of spawners here, we might expect nearly linear increase in the number of smolt out, with some variation around this curve for environmental factors, if our population is depressed. If the population is more at the capacity of the watershed, we could get a full raise of number of the spawners out, and the exit when in the gravel will have no predictive capacity to predict the number of smolts that come out. So we need to understand for each species that we're trying to forecast which point of this curve we are and what we're looking at. And so this is just what a density-independent relationship would look like. In this case, the number of eggs in the gravel might do a good job of predicting the number of smolts that come out, the number of smolts that are entering into that marine environment, which is the start of our one important point of a forecast. However, if the environmental variability around this, the residuals around the time driven by environmental variability is so great, then the number of eggs in the gravel may have no predictive capacity at all. So this is a data set that was collected nearly over 30 years ago on the South Fork Skycomish River by Dave Schuyler. And what he showed the first 6 years that he worked in the system were these data points here. And what he showed was even though he had 2 times as many spawners in some years as other years, there was no predictive capacity. Those spawners couldn't predict how many smolts were going to come out of the South Fork Skycomish. And in fact, if we wanted to predict how many smolts came out of the Skycomish, we'd have to look at other factors, environmental factors. He then experimentally reduced the number of coho that were being put above the fall to see if he could depress the spawners. And when he did this experimental reduction, which you look at the numbers here, this is, down 5,000 spawners down from 15,000 to 30,000 spawners. Something we probably wouldn't do today, but was really a burning question at the time. Could we depress the spawners enough that we would depress this number of smolts out? He found, yes, we could depress them, but, barely. And so coho had this amazing ability for density-dependent compensation in freshwater, such that spawner abundance is rarely something that's going to be predicting the number of smolts out. And we need to be looking, and particularly with this species, at ecological indicators within the freshwater environment. And so right now, when we do this for Puget Sound, the approach that I have applied in the forecast relates to summer low flows, but clearly there's a lot of other variables that could be incorporated there. Rain-derived nutrients coming from the high returns of pink salmon for Puget Sound would be one particular one that comes to mind. Okay, so what happens when we take this and look at its applicability to Chinook? Well, this shows the number of outmigrants per spawners that come out for our different Chinook populations that in Puget Sound, Dungeness, Green River, Cedar River, and Lake Washington, and the Skagit. And you can see in the Dungeness, there's nearly a linear relationship between the number of spawners and the outmigrants. We're nowhere near density dependent in terms of outmigrant production. If it does occur, it's somewhere way off of our current range of spawners. Same in the Green River, there's a fair amount of scatter here, but there's no indication of a nonlinear relationship there. On the Cedar River, when I was initially looking at this data, I thought, oh, it looks like it's flattening out. This is really great. And then we had one really good year of returns of Chinook salmon to the Cedar River, and lo and behold, we get a doubling or more of the number of Chinook fry that are moving out of that system. So kind of put some perspective on it. And the Skagit River, which is almost covered by the reporter panel here, if you look at this kind of pointed from the back, you might start to think this looks like there's some sort of plateauing out here. But if I told you that these were 2 years that had very high flows during the incubation period, you'd realize that there really isn't any plateauing out here. And in fact, using a AIC model comparison of linear and nonlinear relationships here and incorporating the incubation flows, the linear relationship is a much better. The data is best explained by the linear relationship incorporating the incubation flow. So with Chinook outmigrants, there's no indication that density dependence is playing a role, and very important, both the number of eggs that go in the gravel and the environmental variables that are contributing to the scatter around the drying. So that all makes sense, except that Chinook, I think, remember I said, have a lot of life history diversity. And one of the life history diversity, one piece that we see in the Puget Sound is this late time tar outmigration. And these are fish that actually use the fresh water to rear, so they're not just pumping out of there. And this is an example from the Skagit River, but I could show you the same thing from the other systems that were in the previous slide. This is the number of tar outmigrants, these subyearling migrants that use the freshwater habitat to rear as a function of spawners, and there's no relationship. So, life history types may be density dependent even when the total production coming out of freshwater is not. The forecasting methods need to account for these density dependent life stages, especially if the relative survival of the life history types differs from among life history types, and I think Eric mentioned this is one of his key questions for Skagit River Chinook and the life history types in the Skagit River. Okay, so that's a piece of sort of thinking about ecological indicators and stock characteristics in freshwater as they contribute to run size forecasting. And then I'm moving to the marine survival piece here. Unlike freshwater, where we can actually measure smolts in a fair number of situations, we're always predicting marine survival. So the challenge here is either do we have good historical estimates of marine survival of naturally produced fish in a watershed, or are we trying to extrapolate what we know from the neighboring watershed to help us predict something about the watershed that we of interest? So those are the challenges with the marine survival. In Puget Sound, I'm going to focus this a bit on coho marine survival, just because these are where that's where that are available to right now to occur. And we have 3 long-term monitoring stations that have tracked coho marine survival in Puget Sound for over 30 years. Except, well, I guess the Baker is a little bit shorter time than that. It's at 20 years, and then Digby's Creek and Fifth Canal, and Deschutes River in South. And so these are hard numbers. We code a wire tag while smoke's going out. We have an estimate of how many coho were harvested from the quota wire tag, and then we assess the number of quota wire tags that come back to a creek spawning year. So there is less estimation involved in these numbers than a lot of the smolt adult return or marine survival estimates that we may be typically working with. But they do represent regional representation of the Puget Sound basin. So, and there is regional variability in coho marine survival. And this is important where South Sound coho marine survival has pretty much tanked. And although you can't see it here, but there was a 13% in the 2009 return. You can see if you follow Fifth Canal, which are these white squares, we do actually have a pulse of good marine survival in the early 2000s that doesn't track with the decline in the other 2 long-term monitoring stations. So how do we predict marine survival? Well, initially this was set up by looking at a sibling regression, which is what has been talked about this morning. Is jack marine survival being a good predictor of adult marine survival. And so there's 2 big assumptions we make when using this. One is But this would hold up if those early marine conditions were setting the strength of that cohort. Then you should have comparable, then this relationship should be strong. The other major assumption we make is that the maturation rate is the same from year to year. So, like all well-behaved long-term data sets, this one was also well-behaved and was going to disappoint us. And so, in more recent years, since 2000, this relationship has weakened considerably. And this leads to a significant challenge then when trying to develop a solid coho forecast. And it has weakened in that the line has flattened out quite a bit. So the other thing I just wanted to highlight here is that when making these forecasts, it's obviously a good thing to check how good those forecasts are. So if I look just at the big Deep Creek population, so this is the population we're actually monitoring. This is the one-to-one line, and this is the forecasted run size and the actual run size. For most years, this has been a fairly good predictor. These numbers right here occur in those years where the regression line was flattening out. And so those deviations represent that particular issue. However, if we expand this to the management unit, the entire Hood Canal management unit, the relationship is a lot less clear. These are the backward ram runway construction versus the forecasted run size, and there's just fewer years that we have now with that backward slam runway construction available. It's there sort of, but not nearly as strong. And if I were to show you the application of this method to the Snohomish management unit and the Stillaguamish management unit, the relationship would be even less strong, and one might even argue that there isn't very much relationship between what is forecasted and what we see in terms of what actually comes back. So there's clearly issues here that are unresolved and need to be addressed and aren't being addressed with the current method. So my three parting shots here are that life cycle monitoring data can be applied to forecasting in several ways. The need to expand and improve the forecasting tool, especially if we're going to use this for other species besides coho salmon, looking at which life stages are best independent, which ones are not, what's the importance of the starting fauna stock to how we're making our predictions. I think that we need to be considering ecosystem indicators in both freshwater and the marine environment, not just the marine environment, and also for those species that have life history diversity, that's going to be a key component. And the next point is that we can only evaluate our forecast if our run size reconstructions are accurate, and in order to do that, we need good escapement methodologies, and that feeds into the life cycle monitoring framework that is currently in place. And we need to have solid stock ID in mixed stock fisheries in order to do this, and this becomes even more challenging for something like pink or Chinook salmon where we don't have a code-wire tag program, although genetic markers is obviously an alternate way to do that. And finally, the feedback loops of evaluating the forecast are really key if we're going to continue to make steps forward to do it regularly. And that's all I got.
So moving downriver, I'm going to be talking about Estuarine and nearshore population metrics. All right, so I'm going to hope to keep it fairly general and hopefully fairly short. So here's the topic I'm going to be covering. Basically, what I want to show here is first talk about different estuarine habitats that are out there and how we might measure fish within those habitats as metrics, and think carefully about the different species and their reliance on these different estuarine habitats. That would set the context for whether you'd want to use a particular population metric as a forecasting tool for predicting a particular stock. And then I'm going to give two examples. One's pink salmon, and the second is Chinook salmon. Both occur within the Skagit System. And then finally, I'm going to just talk about some of the cross Puget Sound. I don't really know the whole Salish sea for this, but within Puget Sound, the monitoring programs that exist for looking at estuarine fish and the gaps that could potentially be filled. So to start, so here's a little cartoon that shows some of the estuarine habitat that we might see in the Salish Sea. So So starting on this side, so basically what we have is we're moving from shallow water on this side over to deeper water on this side. And so we have a number of systems basically within the rivers, the tidal delta, and smaller creeks. And then as we move this side, we get into the Salish Sea nearshore, and so we have pocket estuaries that Eric was talking about, or just in general lagoons. We have intertidal shoreline habitat, and we have these subtidal neritic habitat. So these boxes up here are sort of places we might expect fish to be. I also want to add in an additional one, this box being the pelagic habitat that is basically most of the volume within the Salish Sea. So how do we measure these? Well, there's a number of different techniques that people have used to estimate the fish population within these. And so here's a number of examples here. So basically within these types of habitats, tidal delta and small creeks, pike nets have been used. Here's an example of a pike net up here, thanks to the Skagit River System Cooperative. And you can also beach stains have been used. Beach stains certainly in the shoreline areas like pocket estuaries and intertidal areas. But in addition, an additional technique that has been used, a visual count. And then as we move into the subtidal, we need bigger boats, bigger nets. And so the work that I've done a lot of with is this surface trawl. Here's an example of a picture right here for sampling subtidal neritic habitats. But in these places also a sort of modified sort of beach stain / purse stain would be a lampar net. I haven't used it too much, but Alan has, so if you're curious about this technique, you can talk to Alan. And then out here, we have big boats and big nets. So you can use big surface trawls or even smaller surface trawls, purse seines, and midwater trawls. And I'm going to save most of this discussion for Rusty to talk. So we have a number of different monitoring techniques, and what we want to do is match sort of the appropriate population metric with the stock. And so what we might sort of do as a first pass is to ask, well, so given that we're concerned about the variability in the population metric, what's the best type of monitoring we might do to capture sort of the greatest range and variability? So if we have a stock that moves through a particular habitat type really quickly, we will not be able to measure that stock very well. And so our measurement would not be a reliable indicator of adult returns. And so here's an example of several different types of measurements for Chinook salmon. And so basically, each of these dots represents the percentage over time that fish were caught with that in that different habitat. So lower river via the outmigrant trap, tidal delta via beach staining, intertidal shoreline via beach staining. No, say something wrong with that. Spike trap, beach staining, and then surface trawl for the subtidal neritic. So for Chinook salmon, at least for wild Chinook salmon, the story is sort of a wash. But all these are well represented. So we might consider looking at all these different metrics for predicting adult returns. It seems that for Chinook salmon, they're using these different habitats fairly extensively. For other stocks or species, we might expect something different. For example, if we were sampling coho salmon, we might expect to see some protracted distribution in the trap, but then very quickly moving through other systems, and so they appear as blips on this graph. And so what I've done here is sort of summarize in a very basic way, sort of if we look at the number of different species across here, number of habitat types here, sort of based on frequency of catch, where would you expect the important habitats or from a predictive standpoint to be? And so this is for hatchery origin fish. For these, we're basically, they're moving through these systems, these nearshore systems pretty quickly. And so the big signal is very much in the subtidal Neritic and pelagic, but they would appear as blips or not at all in these shallower habitats. So when we look at what's natural origin fish, we see something very different. So for Chinook salmon, again, that was an example. As you go across, they are represented very well across those different habitat types. And so all those habitat types might be predictive of adult return. Some salmon might look a little different. Certainly extended residence within these types, but maybe less so within these shallower habitats. Pink salmon, very much not at all here, but into the shoreline, neritic, pelagic, these are places where we see them for extended period of time. And so on. So we see a big distinction here with these three species and these three species, the exception possibly being coho salmon, where I've got these sort of low to medium here, and that really depends upon the life history type you're looking at. If you're looking at the yearling outmigrant, you would expect to see very little signal in these shallower habitats as opposed to deeper habitats. But if you're looking at these sub-yearling outmigrants, those types might become much more important. All right, so I'm going to give a couple of examples of real data and sort of the prediction aspects. One first example is focused on pink salmon using tidal shoreline count. And then the second one would be Chinook salmon using a number of different habitat types. I wish I could be talking about this box right here, but unfortunately our time series right now has sort of some gaps with respect to age structure and other aspects. So I have like 3 data points where we have the early life history and the adult returns lined up. So I'm not going to present that following Martin's comments. So pink salmon. So this is not work that I do, and so I'm relying on the expertise of others in the room, and so Kip Rawson provided this as an example. So basically what people have done is they've used visual counts along specific transects along the shoreline and counted fish at regular intervals in a very standardized method. So basically we have 2 metrics here. One is the sort of mean count over the time period that they're measuring, and then you could do sort of a cumulative abundance, the number of counts and then expanded by the number of days. And so both of these were well predicted. Pink salmon returns quite well, and I mean these linear relationships might not do the pattern justice. For example, here it looks more like a sigmoidal function, so you could probably find a better fit that might actually be theoretically predicted in this case. So this is kind of interesting in that you might expect these counts to be fairly variable, fairly unreliable, but as you add these up and sort of get a sense over time, they become a fairly powerful predictor, at least for the years that we've got so far. So second example, which I'm going to be talking about length, would be Skagit River Chinook salmon. So just a number of people have been talking about it. I just wanted to give a little bit background for those people who don't know. This is the largest source of wild salmon in freshwater in Puget Sound. 6 of the 22 Chinook salmon spawning populations. And then we've got a number of different monitoring activities. Mara talked about some of the trapping data, and then Eric talked about some of the fight trapping data. I'm going to talk about that a little more. But so you have several extensive data sets and good monitoring of adults as well. So one of my initial projects when I started the center was to do some examinations of productivity of the Skagit River population and relate that with some environmental variables. And so we came up with this model, which did pretty well, depending upon whether you were looking at recruits per spawner or spawners per spawner. And we had a number of different variables in here. These latter variables from delta to ocean are basically principal components of several oceanographic factors experienced during the residence period that they were in these different habitats, either the delta, the bay, Skagit Bay, or sort of more marine-like stages. And so we saw so basically I've highlighted those variables in which we sort of have coherence between the spawners per spawner model and the recruits per spawner model. And what we did here was we did a number of different types of model where we varied the parameters, and these are the ones that always kind of popped out, these ones of high importance. So strong negative effect of eggs, suggestive of density dependence, a negative effect of flood recurrence interval, or incubation flood, and then we had this interesting negative effect of the base factor, and a positive effect for this third ocean year factor. So what I want to focus on is this one right here. We found evidence of density dependence, but this model in and of itself, since it focuses on adults, it's not informative about where that density dependence is. And what was interesting was this measurement within the tidal delta showed no real strong effect. So as we moved on to looking at more realistic data, focusing on the data measured in the field, we came up with a more informed story. So I showed some of these slides on Monday, but this is basically the Outmigrant trapping data that Mara has been talking about. And what we see here are basically the counts by life history type as a function of the total number of outmigrants. And the big variable here is fry fish, which move through the system fairly rapidly after hatching. And basically as a main contributor to the variation in the total outmigration size. So those fry are pretty important. They are the life history type, which rears in the tidal delta. So when we think about the link between Chinook salmon and estuary habitats, it's those fry migrants, which are the ones we want to pay attention to. So this is a figure that Eric showed. So basically we have some evidence pretty strong evidence for density dependence. We look at a number of factors. First is sort of the average density measures in tidal delta as a function of the outmigrants. And here's the size of the fish as a function of outmigrants. And basically they increase to a point and they both flatten off, and they both flatten off at pretty much the same level of outmigration. So if there's this apparent capacity, what we'd expect is that when we look at the next life stage, intertidal shoreline, we'd expect to see sort of some increase in the fish that's density dependent, not that's density dependent. So that's in fact what we do see. So basically, this is the cumulative density of fry migrants measured in intertidal shoreline as a function of the density of the tidal delta, and we see with an apparent exponential relationship here. And we've now filled that in with a lot of other data points, and it's a little, the story is still the same, but sort of more, a little more rich in that respect. So we have strong evidence for density dependence and sort of these fry migrants seem to be particularly important in the system. So now, more recently, as I've linked out with some of the adult return data, we can look at two metrics. We can look at marine survival, and we can look at adult returns. And so these are the patterns we have here. So this is a fry caught at the trap. So basically, there's some portion of these fish which are rearing in the tidal delta. But nevertheless, we see these fairly strong relationships. Again, what appears to be a stock-recruit relationship, pretty flat over most of the range in fry levels, and a fairly negative relationship here with respect to marine survival. So these kind of relationships are a good starting point, and what I wanted to do is just sort of think about, well, okay, so if fry are important, so we might measure the life history type itself, or we might measure something about size changes and average size. And so what I wanted to look at was a number of different possible metrics of the fish abundance and their size characteristics. And so that's just sort of as a descriptive starting point of what variables we might consider in an overall model. And so basically looking at a number of habitats and their metrics. So in the river we can look at eggs, fry, and we can express fry in two different ways. Or we can look at all outmigrants. For the tidal delta we could look at cumulative density and then a change in size over that rearing period. And then for the intertidal shoreline, we can look at cumulative density, again, fry migrants, and then size change. And so we see what comes out, we see a lot of strong correlations when we sort of look at these abundance metrics, egg, fry, cumulative density. What seems to be the case is that we sort of, as we move off into deeper water habitats, we get better correlation. The exception being with the tidal delta, which is sort of a subset of the entire population. Also, so when we think about the correlations with marine survival, we see a number of negative effects with respect to different population metrics. Strongly suggestive, again, of density dependence. And we do see some positive effects of size change over the time period within rearing. So this is just a starting point. What we want to do more is think about how to best incorporate these data within the context of a more predictive model. So basically, we're starting out and we'll move from there. So now we have some information about possible measurements we could take within, say, one system. Can we apply that to other systems? And what I did real quickly here, and this is probably got some errors in it, is think about the entire Puget Sound, looking across basins, and thinking about where we have some monitoring. And so basically on this side we have so the different basins on the left, and we have a number of different outmigrant trapping efforts going on that Mara was highlighting, and then these different techniques we could measure. So measurement in the tidal delta, measurement along shorelines or in the subtidal. And some of these I'm not quite sure about, for example, this one right here. Others, for these ones here, well, they're being measured, but they're being measured sort of at the system level, not specific to a particular watershed. But the basic message is we have much more resolution within the outmigrant trapping than we do for any of these estuarine monitoring programs. Particular gaps appear to be in the central basin. And so in sort of thinking about filling those gaps, we might consider that the sort of least expensive strategies would be focused on shoreline monitoring sort of across the board. And with that, I will stop since I'm out of time. Thank you.
Okay, hi. I'd like to thank Correigh and Michael again for inviting us down here this week. And kind of as Correigh alluded to, we're continuing to move out in this sequence of events here, and we're going to take a look at what these juvenile salmon are doing out in the pelagic area of the pelagic Salish Sea. Okay, I thought I'd express this a little bit different than some of the other ones we've seen, and then look at the catch results. And as people have been here all week, and people are here today know very well, we really had a decline in marine survival in both Puget Sound and the Strait of Georgia for coho, Chinook, and apparently steelhead as well. Since really the mid-1980s, a very steep drop down, and then it's kind of stayed at this low level since the mid-90s to now. So we began doing some mid-water trawl surveys in 1998, 1997, 98, to try to understand some of the reasons for this decline and to establish a long-term monitoring program to take a look at some of the variables at sea that we could do. And we were very lucky in these early years, 98, to get together with Dave Beauchamp and his lab and work together with them down here and get a lot of work done. Okay, this is our track line up in the Strait of Georgia. It takes about 8 or 9 days to complete. I'm not going to talk a lot about Strait of Georgia data. I'm going to focus a bit more on Puget Sound. There's our track lines there. We do sets out here in Wandafuke as well, but we're really restricted to the nearshore, to the alongshore area, not nearshore. Traffic lanes in the middle there, and we found stuff on the west coast as well, but I'm going to leave that west coast ocean stuff for Dr. Tidal to talk about tomorrow. Okay, so here's our Puget Sound track line. Generally, we spend a day. The first day we generally spend going south of Seattle, going around Vashon Island and back up, and the second day generally spent going up here back up to Fort Townsend. We have spent a couple of years in both South Sound, a couple of years in Hood Canal. And then just this last year with Correigh, we've gone into Saratoga Passage as well. Just a real brief outline of the survey particulars. The summer survey is sort of the end of June into early mid-July, 8 to 10 days, 30-minute tows. Puget Sound generally right after that. Much shorter time period. Dave and I have been talking this week about perhaps extending that out a few more days. And much shorter tows, the density of the juvenile salmon down here is far greater. And then the same sort of thing in September. That was the earliest we started in relative there. No, in fact, 2004 we were well into October. We don't always get to choose what ship time slot we get. You have to make the best of what you can. And the only other difference between July and September, in July we start at 6 in the morning. Which is about as early as we can convince the ship to start, which is about an hour after it really starts getting light. And then in September we start at 7, where it's still quite dark. So there is that difference there, and we're going to try to keep that apart in some future work. The data we collect on these surveys, species ID for all the fish we catch, we count them, which we use this to calculate some catch per unit effort estimates and some abundance estimates. Collect CWT data for coho and Chinook, do lakes and weights when we can to provide some condition factor and some fork length data. Take some samples. All and scales are pretty routine. Plasma stuff we've collected over the years, and Brian Beckman's going to talk a bit about some work with us, but more with some other groups. In the last couple of years, we've really started doing some multidisciplinary work, collecting DNA samples, RNA, tissues for RNA analysis, and histology. Those of you here earlier this week listened to Christy Miller talk a bit about some of the microsomal or the microarrays she's doing there. We collect diet information, primarily juvenile salmonids. We have done, especially in the early years, a lot of dogfish and hake and some potential predators we were looking at. And then we collect routine collection of oceanographic data as well. Okay, so here's these are Puget Sound data. These are the abundance estimates from July surveys for coho, Chinook, and chum salmon. You see a fair bit of variability. Some good news here, I think, as Laura pointed out in her talk, there was that bit of a blip there at the turn of the decade there where you see some pretty nice survival rates. This survey here in 2000, we kind of maxed out our allowable catch after 3 sets. So we just kind of had to turn around and leave. So that's always going to be a bit of a misnomer. It's probably a little bit high for the short number of sets we have. And then in 2004 here again, a really late survey, and yet there's still a tremendous amount of juvenile chum left in the system that year. So a lot of variability in the data there. And again, fork lengths, the same sort of thing, Puget Sound and Strait of Georgia. I got arrows here for 2007, which was a very dramatic low productivity year in the Strait of Georgia, which we did see in some of the earlier talks this week, was also reflected here in low productivity in the Puget Sound, whereas we saw real impact in fork lengths in the Strait of Georgia, coho, and Chinook. We did not see that in Puget Sound, maybe perhaps in Puget Sound here, but certainly not in the coho data, and chum salmon seem to be unaffected. Fork length is one of the or size, if we want, is one of the main factors in early marine survival. A lot of size selective mortality, and really the main goal of these things when they get out into this pelagic system is to grow. The first 2 to 3 months probably, their main goal is to grow as fast as possible, increase that size, get out of that predation window. And then once you start getting into the fall, into September, they start switching over, and they're really more interested in packing on energy to survive the winter months. Another thing here to note is that the blue bars here are for the non-clip fish. In 1998, when the marine survival of coho had dropped to such low levels, Strait of Georgia management plan started a mass marking of coho salmon, a major clip program, which really benefited us in ways to be able to separate these guys out without having to rely strictly on CWT data. And you see that this non-clip group here has a very strong correlation between fork length and survival rates with the hatchery survival rates. That's the way they do it, or the way we do it. And that is a mix, although it's primarily wild fish. There are, probably 20% of the unclipped hatchery fish in there as well. Whereas the purely clipped group, the relationship is much lower. So the size per se is probably not the key thing here. It's probably growth rate that's really driving the marine survival. Okay, I'm going to talk a little bit about some diet data here. I'm kind of whipping through this stuff because I want to talk about some indicator stuff, but I just want to give some background for the people who haven't been here all week. This is our diet analyst. She's been out with us doing this for 15 years. And basically we just take a subsample of our catch, if it's too big, or the total catch, remove the stomach contents, empty into the petri dish. She estimates some fullness and volume, and then we break it down by percent volume of the items in a diet. We generally do it down to the genus level, the species level, but I'm just going to kind of group them into major categories for today, and I'll make a couple comments about that as we go along. And that allows us to generate these kind of things. So this is for juvenile coho in Puget Sound in our July surveys. As Correigh mentioned, I should mention, this is midwater trawling. We do stratified surveys, so most of our effort is at the surface where juvenile salmon tend to congregate. The juvenile Chinook in particular will filter right down through the water column. So we do fish down deep as well, but I'm not going to get into any of that differences in strata today. The blue bars here are the amphipods, things like damarids and hybrids. The red are juvenile crab larvae, shrimp larvae, primarily crab megalops. The yellow bars here are euphausiid. The white are is a catch-all category for everything from polychaetes and cheated gnats and pteropods and seaweed and rubber hooks and anything that doesn't fit into any other categories. And then, of course, the fish or curios category, which is in both systems primarily juvenile herring and sandlets. And I should point out, in 2003, the Richter was unavailable, so we were unable to get a survey in. And in 2010, I was unavailable, so we were unable to get a survey in. And we generate these things for all 12 years. So what I did for this exercise when I was talking to Correigh in the summertime is try to take a stab at the way Bill does things and generate a correlation matrix, grabbing a bunch of things. I'm kind of just sticking with data from the ship for this part. There's been lots of other talk about abiotic factors here, so I thought I'd just stick to what the fish are telling us. And you grab the few indices that, really lots of time. Grab the few indices that show some strong correlations and try to avoid some of the auto correlations you get there. And rank those out, generate your red, light, green, and rank your average. And we end up with something like this, where we've run the survival rank against that and we generate that. It's not bad for a first stab at it. I'm sure if we put in some abiotic factors in there, that would tighten that up quite a bit. So these are the values that I determined for Strait of Georgia, the fork length, stomach volume in July. The percent of amphipods in the diet in July was negative. The percent of fish in the diet in July was positive. August sea surface temperatures. Obviously, it makes a little sense there. The higher temperatures in August, you're really starting to approach 19, 20 degrees in the surface pelagic water, so you can see clearly how that would be a bit of a negative impact. The March Fraser flow, again, a high flow, high salinity, or dropping the salinity way down. September, negative with decapods, but still positive with fish. In Puget Sound, fork length isn't as important, but we do see condition factor dumping in. Stomach volume is still important. Percent of fish, the September abundance, and the September amphipod. And again, I did kind of run out at lunchtime to kind of grab the Grace Rocks temperature profiles after Nate and earlier talks this week, but I didn't quite have enough. I only have it up to 2002 on my computer here, so I couldn't really throw it in. But I thought that might be fun. I should mention here too, one of the big differences in the diets that we see is the amphipods. In Strait of Georgia, that is dominated by hyperids. In Puget Sound, it's dominated by gammers. Completely different. Okay, and then the same sort of thing for coho. Again, fork length is important, CPU is important, condition factor, surface temperatures in May, March Fraser flows again, CPUs in September, and September stomach volume was actually negative. As I think Correigh mentioned, you try to figure out why some of these things make sense and why these correlations are there. Some of them just don't make sense at this point. We're trying to tease out these relationships as we go along. And so the same thing, if our coho in Puget Sound, the stomach volume was a negative relationship, which you certainly wouldn't expect, given that size and growth rate appear to be so important in driving these things. And the euphausiid volume, September CPE, and September fish volume, but again, didn't grab any abiotic data. Okay, and because it's a workshop and we're allowed to say stupid things, I thought I would throw in a forecast just for fun. So if we run through the data that we got from our surveys this year, this is actually an older, I should have changed this too because I went back and combined them. But basically both the surveys for coho and Puget Sound this year suggest that next year should be a pretty decent return. For Chinook, however, last year and this year's data suggests that there's not going to be any real improvement in the values we've seen. I know for the fish managers, I've certainly heard this morning, this is not the kind of thing they want to hear. They want to hear hard numbers. But that wasn't the goal of our work when we started out. We're just trying to understand these trends and get some underlying issues behind them. And as Mara said, you can't do all this work without lots of help. And these are a lot of the people who've come out over the years. Top couple lines are people from DFO in Nanaimo. Lots of Dave's students and worker bees, although Dave's never quite made it out, but lots of people from DSW, and of course Correigh and Casey were out this year from NOAA, and of course the crew and the officers of the various vessels we've been on. Thanks.
So, Rusty and Mara, join me up here and have a discussion about this.
All right, what I'd like to suggest that we do this time, if you look at your agenda, there's actually a question being posed to you for discussion. And it says, I left it behind with the table. How do we make progress on salmon population metrics? Yes. So why don't we do questions and answers with our speakers for about 10 minutes or so, and then see if we can make progress on that question. Okay, with 10 lights on. Yes. And boy, it looks like you were first. So here you go.
I just had a question for Mara. I was wondering if you had new survival estimates for pink salmon to get some sense whether the recent increase in pink salmon abundance was freshwater or marine.
I cannot put any of that together. That would be possible. Hasn't been a focus.
I suppose this could go to anyone up there, but in particular for Chinook, with the variability of my existing strategies, obviously that creates some challenges for how you parse out these different marine survivals at various stages, and then you add to that the complexity of the age structure. So cohort analysis then is almost something you have to look at across those two different variables. Has there been any sort of effort towards trying to isolate those from one another that you guys have worked on or you're aware of?
Yeah, so I've been trying to assess for at least a couple populations what the contribution of different systems has done. And I think one point that I didn't make within my talk, which is relevant from your standpoint, is that you have a population which enters into the marine system. They're going to experience mortality and variable rates. And so what we want to be able to do is factor that in. If you catch them at a late stage in that mortality has been subsumed. And so from a predictive standpoint, maybe your later cohort is going to predict your returns the best because it's filtered out all the dead fish. But from the perspective of some of these other aspects of management, for example, recovery, that those dead fish represent the potential for that population to be increased. Right? So I would say, to answer your question directly, the data I showed a little bit of really suggests that tri-migrant component for at least some populations within Puget Sound is really needs to be paid attention to, and could be a very straightforward target for them.
I would also add that Mark Goodell and I, my next paper I'm going to write, I'm not sure what he's got in between, but we're going to really tackle that issue, really go into detail into breaking down the system. And also asking again something that Mara mentioned, we're starting to get a lot of good DNA data now. And certainly for us out at the Pelagic, we have no idea where these fish are coming from. So that will really help us to tease apart some of that fuzziness and get down to the stock level approach.
So my question is, given the LL capacity example and a fair amount of data on capacity of estuary habitat, would one of the first steps we could do in terms of management be to try and model what those capacity limits are, and do the salt can occur. We feel like we have enough information to do that in a global sense, so that we go first shot. One of the things we do come out of this meeting is a suggestion everyone measure via GIS their estuary habitat, and then we can apply hectare per hectare numbers to get us a sense of what the capacity is.
Yeah, so I would say that can be a valuable exercise, and particularly from the perspective of recovery management, right, recovery science. So I'm going to raise this as sort of more of a question. I'm skeptical, potentially, whether that, for some of the reasons Mara talked about, is whether that, from a sort of predictive standpoint for sort of looking at returns, whether a sort of capacity estimate for your population is going to be informative. One way it could be, though, is that from a longer-term perspective, is if you're expecting changes in capacity, you'd expect sort of changes in your trend or your average baseline to reflect that. And so in that respect, there's probably some value on a longer-term trend to incorporate something.
So just a follow-up then, what about if we develop a relationship, low, medium, high, to varying spring conditions, if you will, in terms of the productivity out of that capacity? So, for example, in the filling, we measure our capacity at 450,000 smolts. But we know that varies based on salinity and dissolved oxygen. Then I could see how that would help us in terms of predictive return.
Yeah, I could see that as being more valuable.
Just want to add to the discussion of capacity, because I think that a lot of the capacity estimates I have seen have been driven by habitat metrics, which is solid, but now we have fish and habitat metrics that could be put together. So, for example, with the Chinook outmigration, I had didn't show up here, but I had fit the Belgian whole herd to just that far the freshwater bearing component, right? Because you have a plateauing out there in freshwater for that freshwater bearing component of the submarines. And Krista Bartz at NOAA had independently modeled the habitat capacity for Chinook salmon in the same watershed, and we got almost exactly the same range of capacity. So it's nice to test it that way, because now we have specific data to tell us, as well as the habitat data that we want to make some assumptions on physical.
I've got two questions, one for Mara and one for Correigh. So I'll start with Mara first, and once you've answered, I'll ask the question to Correigh. I thought it was really interesting to see the time series of coho survival, and to see where they actually diverge and whatnot. One of the things that I did not see was just a simple bivariate plot of what's the survival of stock A versus stock I can't remember the names of two stocks here, to see to what extent they're strongly correlated. Obviously, there's the one that had the white dot didn't fit very well the other one. But that can give you an idea right away what the scale of the issue is for those particular stocks. So if you have two of the three stocks that are strongly correlated to one another, maybe it's not the potencies that are very local that are important, but it's more perhaps the common pool of the ocean that they encounter that is important, whereas the other one that sticks out might be giving you that indication. So if you haven't done this analysis, I strongly encourage you to do that.
And so I had looked at that a bit, and I looked over the entire time series, the correlation is very poor. If I look at 5-year intervals, the correlation has become tight. So I'm not sure. To me, that speaks a lot of the regional variability and the regional contributions to the marine survival of these Puget Sound coho. But you're absolutely right. It's a great way to sort of kind of tease apart both the predictive ability and then also where the bottlenecks may be or the settings from both sides.
Okay, so that's actually a very important point to make, and so that at least you know what scale you need to look at. So I'm very happy to hear. that you're looking at that. And then this point is for Correigh. I thought that the results you had were also quite interesting, especially with the adult percentage of function of small, you saw feathers in the whole head, hockey stick model there, because that's in the unit. You also have a relationship between marine survival and a number of smolt output or extraction. And also there's a negative relationship between size and smolt. I think these are really cool results. And this brings me back to one of the presentations I had earlier about the potential effect of precipitation on egg scouring in Chinook. And I think if you guys can work together to try to look at the potential impact of these climate change on the number of smolt birds coming out, the size, and the likely survival, and then you can look at consequences for the population.
Unfortunately, Martin and I are in the same program, so we can probably work together. But one of the points, I would say, I think that there are some strong relationships that we've found within the SCAT, the work that Mara has been doing, focusing on sort of those freshwater processes, strongly influence that egg-to-fry survival or egg-to-outmigrant survival. And I think that we now have a pretty strong mechanismal basis there. And what we need now is that second component really. So right now we're working on this assumption that there's these interdependent factors within the estuary and nearshore, and those are what's sort of fixing what's going to happen to the cohort over time. But I think that's a place that could be improved when thinking about this sort of system from a predictive standpoint.
A comment on that negative relationship with the floodplain. With some of the work we're doing in College and Bay with the deep sailing program there, and I think maybe even Correigh mentioned this with the difference of the movements of the wild fish and the hatchery fish, in that the wild fish do tend to spend time in that estuary area, so you see an increase in growth, whereas the hatchery fish tend to go right through. So the size really doesn't vary over several weeks, because the fish just aren't spending time there. It's just the size of the migration.
Are there other questions or comments? Right?
Actually, getting to the question that we're supposed to be addressing, it seems like one way to look at salmon, we kind of know there's a lot of stuff going on, whether it's in the delta tidal areas or the estuary nearshore pelagic. It's one way to look at that as well that integrates across that is how many fish is coming out of Puget Sound or out of the Strait of Georgia. And I guess both to Rusty and Mark, have you guys looked at that from the one standpoint? And do we even know whether most Puget Sound fish are going through the Strait of Georgia or do they go north? We have any respect to which way they're going.
Puget Sound fish?
Yeah.
We rarely get that many Puget Sound fish up in the Strait of Georgia. One of the another of the differences between, particularly for coho, is when we do our work out on the west coast in the summertime, if we get 50 TWTs, 45 of them will be from the Columbia River, and 4 of them will be from Puget Sound or farther. We rarely get Strait of Georgia tags out on the west coast in the summer. Even in September, it's not that common. So the Puget Sound coho is definitely leading the system, before we're really here, and whereas in the Strait of Georgia, I think there's still a substantial population there. So we're kind of mixing that a little bit to get a real number of how many fish are moving out of Puget Sound. I think our numbers for the Strait of Georgia perhaps a bit better.
Oh, yeah, you too. Okay.
Either mark number 1, number 2, or number 3, because we've had all left of us here, so just tell me how it's going to work fine. So for the Chinook and the Puget Sound Chinook, we usually don't see a whole lot of them until the winter off the west coast of Vancouver Island, which suggests to me that they're staying for quite some time, at least within Puget Sound and possibly one Puget Strait. For coho, Rusty is right, at least in terms of the corduroy tag, you can see the corduroy tag coho appearing off the west coast of Vancouver Island as early as the fall. Unfortunately, the DNA on coho at this point is not able to resolve the Puget Sound coho versus the northern BC coho. When we run this on fishes that had tidal tidal, we know what the origin is. It tends to allocate a lot of the Puget Sound and coastal Washington to northern BC. So when we do have fish that are allocated to northern BC off the west coast of the Vancouver, we have no idea if these are actually fish that migrated from the north to the south or they're fish from the southeast into the north. So until this is resolved, I'm not relying too much on the coho either.
Yeah, Kurt and then Alan and others can talk about how to make progress, or what progress we need in the situation.
So this is getting at the question, the question for Mara and Corey. Should we be looking at indicators and making forecasts based on specific life histories within different river systems? So instead of making a Skagit Chinook forecast, should we have a Skagit Fry forecast or a Skagit forecast based on PAR? Is that complicating things? It just seems like those. Okay, Rusty, you can answer.
It's complicated thing.
Yes.
I just want to, I think Corey and what I was putting out there was pointing out some of the complexities here when we have species that have so much legislative diversity. And so what's here is sort of the, hopefully close to the apex of scientific confusion. But in order to actually get that down to a discrete set of advice for forecasting and for management purposes, I don't think we're there yet. We're struggling. We've got to understand the life history, the juvenile life history of the Chinook, where the density-dependent bottlenecks are, how those contribute to return, and having worked out that biology, now we're in a good place to really, provide some clean information for fishery management. If that fits in.
But on the other hand, the more models you have, the better, as they start converging. So it's not a bad idea to track all of those things, and even as far as consolidating things through the different zones we've been talking about.
So one additional point of thinking about it from the perspective of Skagit versus Green River or Mr. Homish, one might expect that these life history types are variable across these different systems and are contributing to the cohort that is in Puget Sound quite variably. And so from that perspective, there might be an argument for focusing on local factors in your prediction model.
I guess my question is more related on how important you feel it is to track these fish as they move out to the ocean. One of the interesting things that Kurt could talk about, or Eric could talk about a little bit more, was the work done in the San Juan, which seems to indicate an absence of fish coming through Admiralty Inlet, passing through the San Juans on their way out, and an abundance of some of the Whitby Bay or Whitby area fish coming north, and the Nooksack fish coming north as well.
From the midwater trawling point of view, it's very difficult for us to fish in these areas. The tidal flux is really high. We've tried many, many times, and it's just, maybe 1 out of 20 times that we actually get a steady. It's just too much flux in there for our risk.
So I can just add a little bit to that now. So certainly my work in the San Juan and West Puget Sound, so what we showed was that Whitby Basin populations were in North Puget Sound and in San Juan. South Sound populations tended not to be in either of those areas. And Canadian fish were in the San Juan, and much of it was a little bit in the north of Puget Sound. So we didn't, we were just focused in San Juan and North Puget Sound, so we kind of really checked the South Sound, Central and South Sound fish went out to Puget Sound proper in terms of that?
We haven't done a lot of work in especially the eastern part of FLT inlet, and we're hoping to get more in the next couple of years.
We're going to move to that question that is on the agenda, and I'd like to clarify that. So we've heard a number of different talks about population metrics, so each of us speaking have identified some gaps, and so thinking about the metrics we mentioned, and thinking about the potential opportunities and gaps out there, sort of, I'm interested to hear and how we might develop a more sort of sophisticated monitoring program with these different subspecies.
Yeah, I guess one of the information needs I've wondered about is, given that you're not going to be able to do the sort of work that Mara is describing in all populations, you're not going to get outmigrant data everywhere. How do you decide what are good indicator populations for what broader groups? You know, like in personnel, the assumption was always that if you pre-indicated what was going on in personnel, and maybe that was a good assumption and maybe it wasn't. But at some point we should be able to maybe more quantitatively approach that question.
My question is just on the question being asked is, we're identifying gaps, but I was also wondering if we shouldn't also flag existing programs, because I know just in terms of funding stability and such, we had a good start with some of the programs that Rusty's involved with, Mark, others. So it's not just a question of gaps, but also hanging on to what we have. So that's another thing I would just add.
Very well taken.
Long-term data.
Other thoughts?
Basically, from your different points of view, given limited resources, is it perhaps more logical to focus more on the adult population and fauna escapement associated with it, and the smolt migration going out, and trying to improve those first, and then move on to some of the more distant activities?
I'm not quite sure on that. In the Strait of Georgia, for example, one of the consequences, or I guess part of the mass marking program, we provide a predictive index, and it's solely based on our CPE of hatchery fish in July, and it's regressed against the marine survival for half of 6 or 7 hatcheries combined together. And we've been doing that since kind of like the regime shift in 2000 when the Sicily model started falling apart. And so we're one of 6 or 7 different models that the coho forecasting team uses. And it's a very simple model, and we can provide that data in July of the ocean entry year. And it performs about as well as all the other models too. So I think there's value to that, even with the simple model. For us, and those of you that were here for the entire week, one of the big knowledge gaps we have, that was mentioned earlier this morning, is the bottom up. What are the zooplankton and ichthyoplankton doing in those first 4 to 6 weeks before ours? It's kind of the gap between the work that these two are doing and what we're doing. So I guess in the light of trying to figure out what progress we need to make on metrics, first 2 days we talked a lot about steelhead. Haven't heard much of a peep about steelhead, so I guess the question would be pretty simple. Do we have comparable population metrics for steelhead in terms of upstream, downstream data, for example?
So I'll answer that, and I actually wanted to, and for you guys, I think you were asking all of us, yeah? Okay. there is less information upstream downstream for steelhead in terms of the length of the time series than for Chinook or coho. Chinook and common steelhead across the time series is probably about the same length. There is one very long-term upstream downstream for steelhead in Puget Sound, that's the Snow Creek data. But the emphasis on steelhead monitoring, juvenile monitoring has increased in the last several years. We now have several tributary tracks on the Skagit River, and we're estimating good solid estimates out of the Quality and Benjamin River, and recently working on the Duckwood River as well. So there's, and I can say, kind of adding in there on my head at the moment is the CSW monitoring. There's some other steelhead estimates of the canal that the very recent group has been making. So I think steelhead information is starting to be built. We're at the beginning of that long-term data set from the perspective of the small monitoring. They're very difficult to estimate because you're so good at avoiding the trap. So you feel that? Okay. And what was Alan's question? Oh, I'm going to go. So you asked Alan when you whether the adult escape and monitoring is the most significant gap with respect to information gap. I have two thoughts on that. I think that there is a lot of work that could be done to improve the adult spawning escapement estimates in Puget Sound, and there are approaches now that are being used on the Oregon coast and the Lower Columbia that could be applied to Puget Sound. We currently are applying a genetic marker capture tool for salmon in a subset of basins to kind of validate, our current methods, how good are they or not. And this whole concept of how do we know our estimate is unbiased and what is the precision of that estimate, that's sort of kicking up right now. And I think there's a lot of work that can be done with Puget Sound stock also in that regard. In terms of applying this, the relevance of that to run-type forecasting, I think that it's one of the key pieces is to understand what environment does that sensitive dependence kick in, because that's going to be the bottleneck. And if that environment is the freshwater egg to smolt trap, like with the coho salmon, for example, then that spawner abundance becomes extra important. If that environment is the estuary, which in many cases the Chinook populations of potentially some populations, although we don't have the data to show that the spawner actively using that estuary, then maybe less. Maybe it's that outmigrant to estuary density relationship that's the most important. So I guess that's where I answer that question.
Going back to Kurt's question about steelhead, people are probably well aware that our ability to measure steelhead in many of these systems is fairly limited with the fresh water, so it's in the surface farm work that I do, is my understanding, resting that is pretty they're not prevalent in the water process,
I could count on one hand and on the steelhead about 50 years.
Right, so for steelhead, we might, assuming we had some predictive adult data for matures, we might not use juvenile metrics in this case for trying to predict this.
Just to try to add to exactly that, and Mark, correct me if I'm wrong, but in addition to being great at trap avoidance, we have a hard time getting our hands on enough wild scopes to get an efficiency for the fish there, because they're not easy to get big numbers of.
Yeah, so there's in order to calibrate the trap and do that marker capture design, you need to have enough fish to mark and release, so you can do the calibration, and you need it throughout the outmigration period, not just once or twice, but as often as you can throughout that outmigration period. And so there are three ways that we've gone about tackling that issue, and it's depends on the system. One is if you can find a slot in the system that has a really solid foul lag, then if you can catch 5-10% of the outmigrant, and there's enough steelhead spawning in the system, then you're able to catch enough fish to do those mark release trials. And so the Nisqually River is one place that we can do that. Another approach to steelhead, actually I think I spoke on, but another approach to steelhead is if you have your marks group comes from an upstream tributary, where you're catching, you put in a fence here, you're catching 100% of the steelhead. Now you can catch 5,000 steelhead coming out of upstream tributary, and now if your trap efficiency is 2, 3, 4%, you have enough fish coming down over the entirety of migration, and that's the other key, right, that you can calibrate your trap for steelhead. Another strategy is don't try to do the biggest portion of the system, but try to get a good estimate from some of the major spawning tributaries. So that's another approach, and the last approach would be to use hatchery steelhead as a surrogate for the wild fish. Releases of 500 to 1,000 hatchery steelhead at paced intervals throughout the outmigration period. There are issues there because those fish have been rearing in concrete climates, and they're bigger, but they also don't have they don't swim the same as a naturally produced fish. And so, of those, that would be my least favorite, but sometimes that's what you got, and so that would be the last one.
Okay, thanks. It's just about 2:30. Any last ideas for how to improve?
I just want to say we use Estuarine and the work on the Estuarine is very effective for steelhead. But that's open water. They're out in the middle of the channel, deep down. And we catch as many steelhead as we do coho and other stuff. So it's really effective. They have a really sharp tooth to them.
Okay. Okay, you guys, I think we should take a break.